Pleasant day to everyone. In behalf of Sir Aristotle Vega, this is Kenneth Frias, and our lesson for today is about cellular respiration. Before we start the lesson, let's take a look at these two cells, the animal cell and the plant cell. If you notice, when you inspect their structures, you can see they have differences and they have similarities. For example, the plant cell has cell wall, and it has the structures called larger vacuoles as compared to the animal cell, which has smaller vacuoles, and the animal cell only has cell membrane. But uh, both of them have membrane-bound organelles, and one of these membrane-bound organelles that is related to our lesson is this, called the mitochondrion. And we will also focus on the cytosol, which is found in the cytoplasm, because these two parts, this organelle called the mitochondrion and the cytoplasm, both play an important role in cellular respiration. If we focus on this organelle mitochondrion, you'll notice that it has similar structure to the chloroplast of the plants, because the mitochondrion has two membranes. So there's the outer membrane. This is the outer membrane. Then there is what we call the inner membrane. So this means that the mitochondrion has two membranes. And between those outer and inner membranes, it's called the inner membrane space. Now, inside the inner membrane, there's a fluid-filled structure here called the matrix. This one. This is the matrix. And just in the inner membrane, you notice that the inner membrane of the mitochondrion is folded. So they contain folds. So these folds are called cristae. So you need to remember these basic parts in the structure of the mitochondrion, because they are very important in the process of cellular respiration, aside from the cytosol, which is located in the cytoplasm. Now, for the lesson, let's define first what is cellular respiration. So, cellular respiration is a process which it breaks down sugar molecule. So, usually it's in the form of glucose, and this glucose acts like a fuel in order for the cell to assemble ATP or adenosine triphosphate molecules. Now, this ATP molecule is very important because it is the one that stores what we call the chemical energy in the form of covalent bonds that can be used by the cell for its cellular activities. This is the general chemical equation, which is a balance reaction of the cellular respiration. So in this, it states that one glucose molecule will combine with six molecules of oxygen. So these are called the reactants. And the product is six molecules of carbon dioxide and six molecules of water. These are the products. And if you notice, if you reverse this reaction, it's what we call the photosynthesis. So cellular respiration is the reverse reaction of photosynthesis. Now, in cellular respiration, remember there are four major stages. In some books, there are three major stages, but here we will discuss the four major stages. So I included the link reaction. So what are these four major stages? First is the glycolysis, and you have to remember it is the very first stage of cellular respiration, whether it's aerobic or anaerobic. So glycolysis happens in the cytosol, which is in your cytoplasm. Then after glycolysis, there's what we call the link reaction, which happens inside the mitochondrion in the matrix. And this is very important because the link reaction from the term itself, it links the glycolysis and the Krebs cycle. So link reaction is important because it processes the products of glycolysis so it can be used in the Krebs cycle. And Krebs cycle is the third stage, which also happens inside the mitochondrion, specifically in the matrix. This Krebs cycle is also known as citric acid cycle or TCA or tricarboxylic acid cycle. And the last stage, which produces lots of ATPs as compared to the previous ones, is what we call the electron transport chain. And this also happens in the mitochondrion, but in the inner folds of the inner membrane called the cristae. As you can see here, the illustration, so glycolysis happens in the cytosol, so it is outside the mitochondrion. And then the next is what we call the link reaction together with Krebs cycle, and then which happens in the matrix, and then in the cristae or the folds of the inner membrane is where the electron transport chain happens. So let's start with the first stage of cellular respiration, which is called glycolysis. Glycolysis, when you look at the term, glyco means sugar and lysis means to break down. So generally, it's a process of breaking down sugar or splitting sugar. So that sugar is in the form of glucose. And it happens or it occurs in the cytoplasm, specifically in the cytosol. So in this process, the simple sugar, which is usually in glucose, and it contains 6 carbons, they are split into two. So 6 divided by 2 is 3. So that means each split molecule contains 3 carbon molecule. Okay, so each molecule contains 3 carbons, and they are called pyruvate. Now, the splitting process of glycolysis requires 2 ATP. It's like an investment, and then it with the help of some enzymes and some nucleotides in the form of NAD plus. So these are the things that are required for the glycolysis to occur. And remember, this process produces a total of 2 net ATPs. So when you say net ATPs, you have to deduct the one that you have used, because when you compute for the gross ATP or the total ATPs produced in glycolysis, it's 4. But you have to subtract the 2 ATPs initially used. So that means glycolysis could produce a total of 2 net ATPs. Now the details we'll discuss on the next slide. So this is how glycolysis happens. So remember, glycolysis is the very first stage of cellular respiration. So it happens in the cytosol, in your cytoplasm of the cell. Now, there are two basic phases happening in glycolysis. First, there is what we call the energy investment phase. What is this energy investment phase? In order for the glucose to be split into two molecules, there is a need for the participation of 2 ATP molecules, as you can see here. So these 2 ATPs are already available before the glycolysis even starts. So what happens here is that the ATPs will give, each ATP will give their phosphate to the glucose, and that ATP will be converted into what we call ADP or adenosine diphosphate, a low energy molecule. And as you can see here, the glucose molecule is transformed into another form. It's called fructose 1,6 biphosphate, but it still contains 6 carbons. If you count this, it's still 6. It only has two phosphates at both ends. And then eventually this fructose 1,6 biphosphate will split with the help of an enzyme called aldolase. And then it will become what we call these two molecules. This is not yet pyruvate. This is what we call G3P and DAP, glyceraldehyde. So this is glyceraldehyde triphosphate, and the other is DAP, also known as dihydroxyacetone phosphate. Now, the details here will be discussed on other subjects in biology, which is more advanced when you discuss biology. But basically, this G3P and DAP each contains three carbons in its molecule. Now, what happens next is that these two molecules, the G3P and DAP, will be further transformed. So, with the participation of these molecules, the NAD. If you remember, NAD is capable of receiving electrons, so it's an electron carrier. What will happen is that the NAD will interact with these two molecules, and what will happen here is that with the help of NAD, they will attach additional phosphate at the ends of this molecule. So this one will attach here. And in the process, they will get electrons. And this process is called oxidation because the molecules lose electron. So what happens here, as you can see in the next illustration, the NAD will become NADH. That means it already carries electron. And then you notice the transformation in these two 3-carbon molecules. And then this is what we call the energy harvesting phase, wherein the glycolysis will already make ATP. So what happens here is that the phosphates that are attached, these phosphates that are attached to these 3-carbon molecules will, so here, that it's not here, it's first in this diagram. So these phosphates here will attach to the ADP molecules that are present in the cytosol. So once they combine the ADP and the phosphate, this ADP will be transformed into ATP. So that's the energy harvesting phase. So in the process of glycolysis, as you see here, there are 4 ATP molecules that could be produced in the process of glycolysis. And as a result of that, the G3P and the DAP before will become what we call pyruvate. So this is the end product of glycolysis. So it produces 4 ATP molecules and then NADH and then pyruvates. Now remember, pyruvate comes from the glucose, which was split a while ago, and it contains 3 carbons. So as the result, the products of glycolysis are 2 net ATPs. So when you say 2 net ATPs, so the 4 that has been produced before, you will subtract the the 2 ATPs that you have used in splitting glucose, so in the energy investment phase. So that means the 2 that will remain is the 2 net ATPs. And then it will also produce 2 pyruvates, which is composed of 3 carbons. So it is important to count the carbons here in this process. And 2 NADH. So this NADH carries what we call the electrons. They are electron carriers. Now, after glycolysis, these 2 pyruvates, so these 2 pyruvates which contain 3 carbons, they have 2 possible pathways. Now, in the case of the absence of oxygen, that those 2 pyruvates will undergo what we call anaerobic respiration, or in other terms, it's called fermentation. That happens if there's no oxygen. And that means the pyruvates will still stay in the cytosol. So in that case, in the absence of oxygen, these pyruvate molecules will be further transformed into 2 possible forms. First is the lactic acid, which is in human cells. This is the one that's being produced in anaerobic respiration, lactic acid. And we usually experience this if you're, let's say, you suddenly engage in a strenuous activity like sports or exercise without proper warm-up. That means there's not enough oxygen being received by your cells. So that means your cells are forced to carry out what we call anaerobic respiration. And the other possibility is that the pyruvate could undergo the transformation into alcohol in the form of ethanol. And this ethanol also produces carbon dioxide. And the purpose of this anaerobic respiration, since there is no presence of oxygen, it just converts the NADH, which carries the electron, back to NAD+. So it's just a way to keep the glycolysis going in the absence of oxygen. But in the presence of oxygen, then aerobic respiration will happen after glycolysis. So this is with oxygen. So that means the pyruvate molecules will then enter the mitochondrion, the matrix of the mitochondrion. So let us assume that there's a presence of oxygen. So we'll proceed to aerobic respiration. But remember, glycolysis is the starting point of respiration, whether it's anaerobic or aerobic. Okay, so in the next stage, Since we're now on aerobic respiration, we assume that there's a presence of oxygen. So after glycolysis, so so this is the illustration here. Glycolysis to pyruvates. If no oxygen, fermentation. So still in the cytosol. And then if there's oxygen, straight to the mitochondrion in the matrix. Now, once there's a presence of oxygen, the pyruvate molecules, remember pyruvate contains 3 carbons, they will enter the matrix of the mitochondrion. So you remember the matrix. Then inside that matrix, the pyruvates will undergo what we call the link reaction or intermediate reaction. Now what happens here is that these 2 pyruvate molecules here will interact with the molecule NAD+. So NAD+ is an electron carrier. And remember, these electron carriers are very important in the succeeding stages of cellular respiration. So what will happen here is that this NAD will split or will remove 1 carbon from each pyruvate, thus reducing the pyruvate into 2 carbon molecule here. And then the carbon that is being split will become what we call carbon dioxide. And you know that carbon dioxide is a waste material. It will be brought out of your body through your respiratory system. And the result of that, the NAD will be transformed into NADH. That means it already has electrons in it. Then this uh, 2 carbon molecule, which previously is pyruvate, will attach to a molecule called coenzyme A, and then it will produce what we call acetyl OA. So here are the products of link reaction. So after the link reaction, so the purpose of link reaction is to prepare the pyruvate to become a form in which it can be used for the next process, which is the Krebs cycle. So the products of link reaction are the following. Since there are 2 pyruvates, it will produce 2 carbon dioxide, which will be brought out of the body as waste material. And then the NADH will just stand by for the succeeding processes that will follow. And then these 2 acetyl-CoA, they are called acetyl-CoA molecules because it's a coenzyme A attached to a 2 carbon molecule. These are the ones that will be used in the next process. So the next stage is what we call the Krebs cycle, also known as Citric Acid Cycle or TCA cycle. By the way, TCA stands for tricarboxylic acid. It's just the IUPAC name of citric acid cycle. And it's also named as Krebs cycle because it's based on the name of the scientist who discovered the mechanism of this cycle, Hans Krebs. Okay, so there are 3 possible names of this cycle. And this also happens inside the mitochondrion in the matrix. So as we have said before, the acetyl-CoA, which is already a prepared form of molecule to undergo Krebs cycle, is in this form. It contains 2 carbons. So I want you to focus here on the number of carbons. So acetyl-CoA has 2 carbons. Then it will combine with the molecule present in the matrix called oxaloacetate, so which is made of 4 carbons. So you just have to add 2 plus 4 is equal to 6. And then the coenzyme A will be released. And this will result in the formation of a new molecule called citric acid. And this is the reason why it's called citric acid cycle. But please take note, in the Krebs cycle or citric acid cycle, although it is called citric acid cycle, you have to remember that the starting and ending molecule in this process is oxaloacetate. So please remember that. But it's still called citric acid cycle because it is the first product produced in the process. So oxaloacetate, which has 4, then you add it to 2 carbons, will produce citric acid or tricarboxylic acid. And then what happens next? So it remember these 6 carbons. Then NAD will interact with this, which will get electron. So that means the citric acid will be oxidized. That means it will lose electron and give it to NAD. And then this NAD will become NADH. And what will happen here is that the NAD will also split or cut one carbon, and that carbon will become carbon dioxide as well as a waste material. Then you notice from six carbon, it will become five carbon molecule. Then the next step, another NADH will interact again and again cut one carbon from this five carbon molecule. So the names of these molecules will be further discussed in senior high school in further studies in biology. So it's just I want you to focus on the number of carbon. So it started with six and then reduced to five. So the result of this is you produce electron carriers, and then from five it will be reduced to four. So what will happen to the one carbon? It will become also carbon dioxide, and thus producing another NADH. And then this four carbon molecule will be further processed, will interact with the following um, substances, the ADP and the inorganic phosphate, which will be transformed into ATP. So that means Krebs cycle is also capable of producing ATP, and then it will also further uh, encounter NAD, and then the cousin of NAD. It's also an electron carrier called FAD or flavine adenine dinucleotide. They will oxidize this four carbon molecule until it will be transformed back into oxaloacetate, which still has four carbons. So if you notice the pattern in Krebs cycle, 4 plus 2 is 6, and then you cut one carbon will become 5, then that carbon will be released as carbon dioxide, and it will be reduced to 4, and so on. Okay, so that means the Krebs cycle will start with oxaloacetate and end as oxaloacetate. But the first product that is produced is citric acid, and it's the reason it's why it's called citric acid cycle. So as an overview of that Krebs cycle, so that we could have you could have a better grasp. So remember, from the link reaction, you produced two acetyl CoA molecules. So remember two, and then it will combine with oxaloacetate, which has four carbons. So it will produce citric acid, which has six carbons, and then it will interact with NAD, which will produce carbon dioxide and NADH. So that will reduce the six carbon to five, and then this five carbon will further react to NAD and will be further reduced into four carbon, and that one carbon that is being split will also become carbon dioxide, and then it will this four carbon molecule will undergo further processing or reconfiguration of its structure. So it will interact with ADP so that the Krebs cycle will create ATP. Remember, this is the the result of these cycles is production of ATPs. And then furthermore, it will still react with another electron carrier called FAD or flavine adenine dinucleotide, and it will become FADH2 or flavine adenine dinucleotide hydrogenase. And then another with NAD, which will become NADH, and then this four carbon will be transformed back into oxaloacetate. So that is the Krebs cycle. So if you're going to get the bigger picture here, it's just you add 2 plus 4 becomes 6, and then 6 will be reduced to 5, and then 4. So that's a cycle. So that's the general process of Krebs cycle. But if for the details, it has a different lesson, a little more advanced biology, cellular respiration in biology. Now remember, since there are in one glucose molecule, you produce two pyruvates and two acetyl CoA. That means citric acid cycle happens two times. Okay, remember that. So one citric acid cycle for each acetyl CoA molecules. So after two citric acid cycles, these are the products. A total of four carbon dioxide, six NADH, so six electron transporters in the form of NADH, and two FADH2. and then 2 ATPs. If you notice, in glycolysis, you already produce 2 ATPs. In the link reaction, there's no ATP produced. In the Krebs cycle, you still produce 2 additional ATPs. Now, you notice that the cell is already capable of producing ATPs, but this quantity is not enough. So it needs another process. Now, remember, this NADH and FADH2, which are electron carriers, they are very important because they will proceed to the next part of the mitochondrion called the cristae, or the inner membrane, the inner folds in the inner membrane, for the next process, which is popularly known as electron transport chain. But actually, this is technically known as oxidative phosphorylation because it's the process of combining phosphorus in organic phosphate to ADP to make ATPs. And there's also an additional or additional process here aside from electron transport chain. It's also called chemosmosis. Okay, so if this is the mitochondrion, after the Krebs cycle, which happens here in the matrix, the electron carriers together with the oxygen will proceed to the folds, the cristae. If you zoom this cristae, you will see several structures here. So it's similar to the light-dependent reaction in photosynthesis. There are also structures in the membrane, in the inner membrane. So if in photosynthesis is in the thylakoid membrane, here in cellular respiration in the mitochondrion, it's found in the cristae or the inner membrane of the mitochondrion. And remember, the region that separates the inner from the outer membrane is called the intermembrane space. And this is also very important in the process. Now, here, this is the matrix. Now, what are these structures? So these are proteins. So there are certain names for that. So I'm not requiring you to memorize, but you have to be familiar. These are called protein complexes. So this is what will these are the structures that will participate in the process called electron transport chain and happens in the cristae. So they have their names: NADH dehydrogenase, ubiquinone, cytochrome BC1, etc. But you can simply name this as protein complex 1, and this is protein complex 2, this is protein complex 3 and 4. And this fifth one is the ATP synthase. You're familiar with the ATP synthase. It is capable of producing ATP when it turns like a turbine. Now, these structures here, the ubiquinone and the cytochrome C, they are electron transporters. So they are capable of fetching electrons from one protein complex to the next protein complex. So, so that's what you need to remember. Now, how does electron transport chain, uh, how does, what's its mechanism? How does it happen? So there are, remember, electron transport chain is a simultaneous event. There are lots of events happening at the same time. So let's try to get into it. So electron transport chain. So these are the first things that will happen. First, remember there are two electron carriers, the NADH and the FADH2. What will happen is that they will go to this protein complex 1. So NADH will go to protein complex 1 and FADH2 will go to protein complex 2. And what will they do is that they will donate their electrons there. So they will donate their electrons. So what will happen to NADH? It will be transformed back to NAD, and the FADH2 will be transformed back to FAD. Okay? So NADH will become NAD, FADH2 will become FAD. So what happens there is when the electrons are being donated to these protein complexes here, the electron transporters will fetch them. So as you can see here, this electron transporter has already fetched, and it will bring the electrons from one protein complex and to the other. And so is this electron transporter. It will also fetch and transport the electrons from one protein complex to another along the cristae or the inner membrane. And that's the reason why it's called electron transport chain. So it's like a chain of events. Now, as a result of that, while transferring electrons, there is an attraction happening, like in photosynthesis. If you see the arrow, the hydrogen ions or the protons that are present in the matrix are attracted and will go towards the intermembrane space here. So that means there is an accumulation or an increase in amount of protons in the intermembrane space. So these protons are also known as hydrogen cations. And due to that, it will increase the positive concentration gradient or the charge inside the intermembrane space. And if that happens, the accumulation of this concentration gradient will force a process called chemiosmosis. If you remember, osmosis is the movement of water through a semi-permeable membrane. But since this is chemiosmosis, it's not water that's moving through a membrane. It's the hydrogen ion. So these hydrogen ions will undergo a process of chemiosmosis, and it will pass through the ATP synthase. So remember the ATP synthase. Once hydrogen ions passes through the ATP synthase, this ATP synthase will turn like a turbine, and it will carry out a process called oxidative phosphorylation. So oxidative phosphorylation is a process wherein the ATP synthase is activated, and then the ADP, which is present in the matrix, will combine with inorganic phosphate, and that process is called oxidative phosphorylation. And thus, this ADP plus inorganic phosphate will produce what we call an energy molecule, ATP. So that's how it goes. So as the electron transport chain is happening due to the donation of the electrons from electron carriers, the electrons are transported by transporters through the process of electron transport chain. Therefore, it increases the movement of ions in the intermembrane space, thus increasing the concentration gradient, and it leads to a process of chemiosmosis, which brings the hydrogen ions back to the matrix through the ATP synthase, and in turn, the ATP synthase produces ATP. But as this happens, you will notice that the electrons that are being fetched by the electron transporters will accumulate in this protein complex. So this is a protein complex 1, 2, 1, 2, 3. Protein complex 4. So in protein complex 4, the electrons will accumulate there. Something must become an electron acceptor, and that's where the oxygen comes in. So this is the importance of aerobic respiration. The presence of oxygen molecule, this oxygen molecule will act as the final electron acceptor in the process of electron transport chain. So these electrons here will combine with this oxygen molecule together with hydrogen ions, and this oxygen molecule will be transformed into water molecule. And water molecule is one of the products of cellular respiration. So that means one oxygen molecule plus four hydrogen ions plus four electrons will produce two molecules of water. So that means as lots of ATPs are produced through the process of oxidative phosphorylation, water is also produced because of the action of the uh, accepting the oxygen molecules accepting the accumulated electrons in the electron transport chain. And because of this, the total the accumulation or the simultaneous events that are happening in the electron transport chain, it produces about 32 to 34 ATPs, or it depends on the literature. Some say 34, 36, 32, but it's about 32 to 34 ATPs and about 6 molecules of water. That's per glucose molecule that is being processed in cellular respiration. Okay, so it still depends on the number of glucose. So the more glucose, you just have to multiply. Okay, so this is per glucose molecule. So that is the product of the electron transport chain. And if you notice, as compared to the previous stages, the electron transport chain is capable of producing large quantities of ATPs due to the action of these electron carriers, NADH and FADH2. Now, as an overview of the overall aerobic cellular respiration process, we're talking about aerobic because we assume that there's a presence of oxygen. So once you eat a food and it's being fully digested, So that carbohydrates will be broken down into smaller molecules called glucose. It will enter your cell, and once it is in your cytoplasm, in your cytosol, then the glycolysis will happen. So what happens in glycolysis? It will split that glucose molecule, or it will split that sugar, and in the process it produces two net ATPs. Now if there's a presence of oxygen, it will undergo what we call ring reaction, or it's also known as oxidation of pyruvates. So remember, glycolysis produces two ATPs, and it will also split the glucose into two, and you call these two molecules pyruvates. And these pyruvates will be processed in link reaction here, and then once it's transformed into acetyl CoA, it will undergo what we call citric acid cycle, which will further process this acetyl CoA into carbon dioxide, which will be released out of the body, and then in the process it will produce two ATPs. Now after that, the electron carriers NADH and FADH2 that are produced in the citric acid cycle will proceed to the cristae, to the folds here, which contains the protein complexes and the ATP synthase, and they will perform what they call the electron transport chain, which will produce at about 32, more or less 32 ATPs. So that means of all the three stages, it is the electron transport chain that is capable of producing lots of ATPs. Now, how is that computed? So there are different literatures available, but we have this as an example. So how is that computation of ATPs carried out, and how did they figure out the number of ATPs produced in ETC, or electron transport chain? So based on experiments or studies by researchers, they have computed that about four hydrogen ions. So remember, the hydrogen ions are the ones that are passing through the ATP synthase through chemismosis. Four hydrogen ions are needed to produce one ATP. So that means the four hydrogen ions needs to enter the ATP synthase to produce one ATP. And also based on their studies, one NADH, that electron carrier, can produce up to three ATPs. Okay, so the details here will be discussed in more advanced biology topics. And one FADH2 can produce about two ATPs. So if we're going to tally the molecules being produced in each stage of the cellular respiration, so it's remember, this is per glucose molecule, so we assume it's one glucose molecule. So in the process of glycolysis, two net ATPs are produced, and two NADH, FADH2 is zero. Now, the next stage, the link reaction, no ATP is produced, then but it's able to produce two NADH and no FADH2. In the Krebs cycle, it is able to generate two ATPs, six NADH, and two FADH2. So if we get the total ATPs produced in the first three stages are four net ATPs. Remember, we already deducted the two ATPs used in the energy investment phase. And when we compute the total NADH produced in the three stages, total of 10 NADH. And then FADH2, a total of 2 FADH2 molecules. So we are talking here about the products produced per glucose molecule. Then we have to compute. Since one NADH is capable of producing about 3 ATPs, and there are 10 NADH produced, so 10 multiplied by 3 is 30 ATPs. Then, since there are 2 FADH2 molecules produced in the three first stages, so in the electron transport chain, this 2 FADH multiplied by 2 is 4. So 30 plus 4, so there are about 34 ATPs that are produced from oxidative phosphorylation, or in the electron transport chain. Plus you add the 4 net ATPs, okay, that are produced in the previous cycles, the glycolysis and the Krebs cycle, a total of a maximum, or about 38 ATPs are produced in the overall process of cellular respiration, specifically aerobic cellular respiration. So the summary of the chemical reaction, as we have shown a while ago in the previous slides. So one glucose molecule together with six molecules of oxygen will produce six molecules of carbon dioxide as waste material, and then six water molecules. It's happening. Remember, it happened in the electron transport chain, and about and due to that overall process, it's able to assemble about 38 ATP molecules. So that's how the computation of ATPs are done. Now, remember that these are just estimates because these are based on experiments. So different literatures have different recommendations, or or they have different discussions about this. Some literature say that there's a reduction in ATPs because I've read some that in order for the ATP to go out of the mitochondrion and be used by the cell, there is it there is a need for a consumption of two ATP, so that will reduce 38 to 36. So it really depends on the literature or the references. So but it's about 30 plus 32, 34, 36, or 38, maximum of 38 ATPs are produced in the process. Now, as a summary of cellular respiration, you have to remember that cellular respiration is breaking down of sugar to make ATP. Now, cellular respiration always starts with glycolysis, whether there's presence or absence of oxygen. Glycolysis is the starting point. And glycolysis happens in the cytosol, which is the fluid in your cytoplasm. Now, in the case of the absence of oxygen, these pyruvates that are produced in glycolysis, they remain in the cytosol, and it will undergo what we call anaerobic respiration, also known as fermentation. And that's the reason why when in making bread, it's there's a fermentation involved there because there is ethanol produced and carbon dioxide produced. So that's fermentation. In humans, we have what we call lactic acid formation. But with the presence of oxygen, these pyruvates, which are produced in the glycolysis, will proceed to the matrix so that it could undergo what we call the link reaction, which will prepare the pyruvates to become acetyl CoA and used in Krebs cycle, also known as citric acid cycle or TCA cycle. Now, next, the electron carriers, the NADH and the FADH2 are very, very important because these electron carriers, together with oxygen, will proceed to the cristae or the inner membrane folds of the mitochondrion for them to perform what we call the electron transport chain. And this electron transport chain will result in the following events: the increase in concentration gradient, then chemiosmosis, and oxidative phosphorylation. So this oxidative phosphorylation will lead to production of lots of ATPs. And last, as a general balance reaction, so that means one glucose molecule needs six molecules of oxygen so that it could process the cellular respiration properly and produce six molecules of carbon dioxide, six molecules of water, and about 36 up to 38 ATPs or adenosine triphosphates, which are energy molecules used by your cells to perform cellular activities. So I hope you understand the lesson, and that will be all. Thank you very much.